So gene flow is the movement of genes from one population or species to another population or species. And it's a natural process. So for example, modern humans have bits of Neanderthal DNA in them. And presumably from some ancient human Neanderthal mating. And mating is by far the most common mechanism for gene flow.
So the focus of the research is mostly about preventing this spillover. This gene flow can be measured by looking at genetic markers in specific populations and its similarity with other populations for comparison.
So that's common between populations of the same species, and well known but much rarer between closely related species that can sometimes form fertile hybrids, so presumably the Neanderthal human example. Gene flow between unrelated species is incredibly rare.
Although rare, there could be some negative consequences to the ecosystem as a result of this changing phenotype. For example, if you're trying to reduce population of an invasive species in an area using gene drive, because of this spillover, it can affect population of related but local species.
So if it's not intended to get into another population, and it does, what's the consequence? Well, for most gene drive designs, there's going to be a rather limited consequence, so that you might be able to detect it by molecular methods, but not see any big effect. But again, this varies from one type to another. Most of these designs present at low frequency, so very rare migration, will have negligible effect. It's only if they can work to spread themselves from that low level, which some gene drives are designed to do and some are not, that there could be any noticeable or measurable consequence in most cases. So again, case-by-case assessment.
So scientists have done and are doing some lab experiments to check specificity of the gene drive. They're working on overcoming this issue, and it can be done by well-planned construction of gene drive. And we can use what is called as a precision drive, where one can use very specific and unique sequences to target the gene in specific species. This will avoid the spillover into related species due to dissimilarity in sequences. For that, we need to know the genome sequence of the target species and its relatedness to other species. So with this technique, the chance of gene flow into non-target species becomes negligible.